Hi class, it's Dr. Lindner. On this next run, we are going to be talking about the neuroglial cells. Now, we've already talked about neurons. Remember, we said we have two types of cells. We had the neuron, which was electrically excitable, and we spent some time talking about the different parts of the neuron, like the dendrites, the cell body, the nucleus, the nissl bodies, the axon hillock, the axon, the axon terminal, the synaptic end bulbs, um, the axon collateral, the Schwann cell, the nodes of Ranvier, right? All these things you'll be studying and just reviewing and looking them over. That was electrically excitable. Now we're talking about the second type of cell called neuroglial cells. Neuroglial or neuroglial cells. It means nerve glue. We have 6 different ones, 4 in the CNS and 2 in the PNS. What does CNS stand for? Central neural system. What does PNS stand for? Peripheral neural system. Now, the neuroglial cells, if you look at this picture, this is a neuron, and everything around it are going to be neuroglial cells. So the neuroglial cells make up half the volume of the central neural system. They're smaller than neurons, but they're 50 times more numerous. In the central neural system, we have 4 biggies: astrocytes, oligodendrocytes, microglial cells, and ependymal cells. We'll go over some of the key functions of them. And then 2 types in the peripheral neural system: Schwann cells and satellite cells. Okay? And of the 2, we'll focus a little bit more on the Schwann cells. And when we look at the neuroglial cells here, we could see a combination of all the different ones combined, right? So here is an oligodendrocyte. Here's a microglial cell. Well, here's a neuron. Okay. Here's an astrocyte. Here's an ependymal cell. These are the ependymal cells. They have some microvilli and cilia coming off of that. You remember what those are. Um, Here is an axon. We have myelin sheath protecting it. There's the node of Ranvier. We have oligodendrocyte, and here's another astrocyte, and here's some more neurons. Okay, so let's look at the astrocyte first. When we look at astrocyte, we see astro, like astrology, and astro means star, and astrocytes are star-shaped. And it happens to make and be the connection. Here is a process of a neuron, and here's a blood vessel. So it makes up what we refer to as the BBB, which is the blood-brain barrier. It forms the blood-brain barrier. It's the connection. It's involved in metabolizing neurotransmitters, regulating the balance of potassium and sodium that are involved in nerve conduction. So that's the astrocyte, the blood-brain barrier. Now, the oligodendrocyte is one of the most common glial cells. It's very, very similar to the Schwann cell. Remember, the Schwann cell was in the PNS, but the oligodendrocyte is in the CNS. What they do is they both produce myelin. And this is like real estate: location, location, location. The oligodendrocytes are going to produce myelin, but in the CNS, whereas the Schwann cell produces myelin in the PNS. So they both myelinate, except the oligodendrocytes myelinate the brain and the spinal cord, and the Schwann cells myelinate everything in the periphery. And this is just showing the process of myelination. Here is a Schwann cell. We can see the Schwann cell, and we can see an axon, and it almost looks like a macrophage, like it's phagocytic, where we see this Schwann cell engulfing, like eating the axon, and it keeps going around and going around and going around, and it keeps kind of circulating, going around and around and around, till eventually we have this really, really nice myelin sheath of protection, protecting the axon. And the space um, between each Schwann cell, remember, is called the node of Ranvier. It's the unmyelinated section. Okay? Microglial cells, it's a phagocytic cell. It kind of eats and breaks down foreign debris, microbes, cellular debris. It's phagocytic. It's derived from cells that gave rise to macrophages and monocytes. Okay? A microglial cell started as a monocyte, which is a white blood cell. A monocyte is a white blood cell that circulates in your blood vessels, and as soon as it finds some sort of lesion or toxin or infection or debris, that monocyte leaves, goes into the neural tissue, and now it becomes a macrophage. But the type of macrophage that it's referred to when it's in the neural system is called a microglial cell. When a monocyte is in circulation and it leaves the blood vessels and enter the lungs, it's called an alveolar dust cell. When this monocyte engulfs damaged cholesterol, it becomes a macrophage called a foam cell. When the monocyte dives into the liver to clear out debris, it's called a Kupffer cell, right? So monocytes change names when they become macrophages, and those macrophages get into different tissues. The name of the macrophage just changes names, okay? Ependymal cells, these are really important. They're going to line the ventricles of the brain, and the ventricles are these cerebral cavities that produce CSF. CSF stands for cerebral spinal fluid. Ependymal cells line the ventricles in the brain, and we have 4 ventricles of the brain, and we'll go over the ventricles when I cover the brain. They line the ventricles, they produce cerebral spinal fluid, and it helps to bathe the brain and buoy the brain against like TBI, traumatic brain injuries and stuff. Okay, it also carries some nutrients as well. Satellite cells, these are just supporting neurons in the peripheral neural system. Okay, they're just supporting like cells. Our Schwann cells, these are the myelinators in the peripheral neural system. And we know about the autoimmune condition that attacks the Schwann cells or that attacks the oligodendrocytes. Once it breaks down the myelin, it starts to negatively impact nerve conduction. And the name of that autoimmune condition is multiple sclerosis, or MS. Typically, MS, when it starts to affect first the eyes, and people get kind of visual disturbances because the optic nerve is kind of small and it starts to target the small nerves first. When we look at the spinal cord on the left and we look at a cross section of the brain on the right, we're going to be looking at just the difference of white matter and gray matter. When we look on the left at the butterfly or the letter H, that's gray matter. So this is all gray. And what's around it in the periphery is white matter. So white matter is myelinated, gray matter is unmyelinated. But look what happens in the brain. It does a flip-flop where we have the gray matter on the outside and we have the white matter on the inside. It does a complete flip-flop. So when we look at the cross section, this is the vertebral body of a vertebrae. These are the transverse processes of the vertebrae. This is the spinous process of the vertebrae. And these are muscles of the vertebrae. Here's the spinal cord, and these are nerve roots. When we look at the spinal cord, we see the highlighted butterfly in the middle. Well, that's gray matter. When we look at what surrounds that, that's the white matter. That's myelinated. Okay? Okay, good place to take a break. When we come back, we'll get into the brain.